Okay, so chapter 14 is about molecular genetic analysis and biotechnology. I'm going to be honest, what's in this chapter is like drinking water from a fire hose. This is like every technique that you would learn each one could be an entire course in of itself. So I'm going to try to cover these techniques in a way that I'm just trying to present what the basic concepts of the techniques are, so you get a sense of how we manipulate genetic material in order to solve human problems and answer questions. Some of the techniques we will actually perform in lab. And so those will be more focused on in lab, less focused on in my lecture. So rely on your practice questions quite a bit after you've done the chapter, or even before you've done the chapter. Look at the practice questions so you kind of get a sense of how much detail I'm asking you to know. But really, the name of the technique and generally what you can get out of that technique is a good way to start as you're thinking about this. So the vignette for this chapter is about Duchenne-type muscular dystrophy. They've been developing gene editing, CRISPR technology as a potential cure for this disorder, and so you'll read about that. A lot of the important work on this has actually been happening at UT Southwestern. Eric Olson is a very prominent researcher in this area and quite famous in this area, and actually he was one of my mentors, and he's a good guy. In any case, what I added to this lecture is this timeline. So I'm going to go through this. It's going to seem a little overwhelming at first, so don't panic. I'm going to go through this. These are what I call the molecular genetic techniques that changed everything. So these are all ultimately Nobel Prize winning inventions and discoveries starting in the 1960s. You'll recall that Watson and Crick only discovered the structure of DNA in 1953. And so at that time, they were still trying to figure out, you might recall in 1958, they figured out how a cell replicates its DNA, Meselson and Stahl, and the semi-conservative replication. And so, in the late 50s, we were still trying to figure out just, how DNA worked naturally in the cell, how the cell replicated DNA, how the cell expressed genes. And so the manipulation of DNA was the next obvious thing to attack, but we didn't have any way of getting an accessible sized piece of DNA. Like if you have a whole chromosome, even from a bacteria, it's quite a huge piece of DNA. And so there was a need to somehow break it up into pieces, and different techniques were tried. But in the 1960s, two scientists, Herb Boyer and Stan Cohen, figured out that bacteria contain naturally occurring enzymes called restriction enzymes, and they could be purified and used to cut the DNA in predictable ways. And it created the ability then to cut the DNA down into smaller pieces that could be manageable. So the use of restriction enzymes was the beginning of the ability to manipulate DNA, because we could get smaller fragments that we could manipulate much more easily. And so that was key. In 1974, the first transgenic mouse was created, and they used, because of restriction enzymes, they were able to manipulate a viral chromosome with some genes. And so I point out the mouse. There was actually some transgenic frogs that were created before that. But since the mouse is one of our model organisms, 1974 was when the first transgenic mouse. Transgenic means a gene that doesn't normally exist in that organism is introduced into the organism. So it's called a transgene. A transgene has to be typically, at least in the 70s, it had to be carried by another piece of DNA, in this case, a viral DNA. And so a transgene would be carried into a cell by a virus, and then the cell would take in that gene, hopefully. So at first, this was, not very successful. I mean, they might get one mouse here and there. It was, not very efficient, but they kept working on it. And like any technology, they make improvements. It becomes more efficient. They come up with little tricks that make it better. And so all of these techniques started off really clunky and not very good, and then they get refined and improved over the years. So in 1977, Fred Sanger created a biochemical technique for determining the nucleotide sequence on any strand of DNA. And we called that Sanger sequencing. It's the very first type of sequencing that was invented. And it involved, at first, radioactivity. In fact, the first 3 genes I cloned, we did the Sanger sequencing with the radioactivity. I mean, it's, it's kind of hazardous. So Sanger sequencing, though, it allowed us to figure out the DNA sequence of any fragment of DNA. And because of restriction enzymes, we could break up chromosomes and then sequence the DNA. And so now we had a technique for that. And like I said, at first it was messy and kind of awful, but it improved over the years, as you're going to see. In 1980, they improved on the transgenic mouse techniques. They invented a needle that could inject DNA directly into an egg, either usually a fertilized egg is what they would use, so really a zygote. But that was an improvement on the transgenic mouse that they had created using a virus. So this way they had a needle that could inject DNA directly into the nucleus of an egg. That needle is so fragile, you cannot believe it, and you have to look at it under a microscope. I mean, they still use that today. In the 1980s, there were extensive random mutagenesis experiments by some Nobel Prize winning scientists in Germany who used Drosophila as their model organism, and it really started the whole era of what we call reverse genetics, intentionally creating mutants and then studying the genetics, the genes of the mutants to figure out what genes were causing certain phenotypes. At the same time, you have people trying to figure out site-directed mutagenesis techniques, because as we've discussed, random mutagenesis is not very efficient. So if we could target, somehow create mutations in locations that we directed the mutation to happen at a certain place, that would be much more efficient. All of the site-directed mutagenesis techniques that were developed, like you can guess, were not very efficient. They were kind of hard to do, but this was the beginning of this, this technology. In 1985, Kary Mullis invented something called PCR, the polymerase chain reaction. It's a method for synthesizing large amounts, that's a typo there, large amounts of a target DNA sequence from small amounts of a template. It's like DNA replication, but instead of copying the whole genome, you're just copying one target sequence and you're copying it millions and millions of times. So you can make a large amount of a target DNA sequence from small amounts of template. This had implications for all kinds of things, but one thing that we're going to look at in lab is DNA fingerprinting. So being able to do a DNA test, being able to do a forensics type DNA test, being able to do a paternity test, that started with the invention of PCR. Before that, it was hard to get large amounts of DNA very easily. In the 1990s, that was the beginning of the genome projects. So they were still using the Sanger sequencing, but there were some improvements on the Sanger sequencing. They had been able to replace the radioactive materials with fluorescent materials, so they weren't dangerous. And the way that the sequencing was done was a little more streamlined and a little more computerized. Still considered nowadays old-fashioned type of sequencing, but the genome projects, especially the Human Genome Project, which we're going to talk about in Chapter 15, began in the 1990s. Also in the 1990s, they started trials on gene therapy. And so gene therapy began. There was one case, one patient that was cured of their genetic disorder in the 1990s. So that was the very beginning. Again, everything going very slowly, nothing, working very well, but everyone's working to improve on this. By 2001, the Human Genome Project was declared finished. I put air quotes there. We're going to talk about that in chapter 15. At least it was as finished as the current sequencing techniques could achieve. We couldn't get past, there were certain things we just couldn't solve in the genome, but we said it's as finished as it can be right now. But as you can imagine, people were trying to invent new types of sequencing that could work around some of the problems that Sanger sequencing had. And these were just biochemical problems, mostly related to repeat sequences. And so in 2001, what's now called next-gen sequencing or Illumina sequencing was invented. So we'll talk about that technique a little bit. It's also going to give you an ATGC sequence of the nucleotides on any DNA strand. It's just it's a different concept, but it generates the same result. In 2011, another company invented something, we call it PacBio sequencing, because the name of the company is Pacific Biosciences. And sometimes this is called NextGen 2.0. Nowadays, we use a combination of these types of sequencing. So sometimes you'll see the Illumina sequencing used or the PacBio. They're both pretty commonly used. Sanger sequencing, I'm sure it still exists. It doesn't cost very much, so it's kind of okay for something you don't need to do a lot of sequencing, but most of the time you're going to have one of the next-gen sequencing. And a lot of universities have now a whole lab that all they do is sequence as a service to the other labs in the university. So that exists. In 2012, CRISPR was first used for gene editing, or shown that it could be used for gene editing. CRISPR is a naturally occurring system in bacteria, but then it's been created as a technique for gene editing. And in the 2020s, I wrote, the Human Genome Project has been declared really finished, really truly finished, because they applied the Illumina and PacBio sequencing methods and were able to really finish even the repeat sequences. So there's a lot here. Overall, what you're really looking at is the ability, the process of improving the ability to isolate and manage DNA fragments, the ability to determine the nucleotide sequence of those fragments, and then the ability to deliver certain genes and certain things into cells and organisms. And those are the things that have been improving over this course of 60 years. So that's an overview. I know that's a lot, and it's might be a little overwhelming, but this is a good introduction to this chapter.